But first of all, I'd like to thank organizers for giving me this great opportunity. So, as Suresh Ratan already mentioned this morning, hormesis means that very low dose of toxin and other stressors may trigger an organism to the opposite effect to the very high dose. So as you can see here, with high dose, it inhibits the organism, but at the very low dose, it may stimulate the organism. So here's the outline of my talk today. I'd like to briefly mention about the aging, especially aging study in Korea, and talk about genome hormesis and radio hormesis. And if time permits, I'd like to briefly talk about next year's IAGG. So introduction to aging. So everyone ages. So as you can see here, you can see the aging of Charles Darwin. But everything has exception. So there are few exceptions who doesn't look aging at all. So she's French actress Isabelle Adjani, and as you can see here, she looks younger and younger as time goes by. And actually, she was born in 1956, and she's quite old lady, and she has grandsons. So here's the advertisement in South Korea. So this picture of the subway, and it's imaginary picture. So right now, it is reserved seat. So as you can see here, it's reserved seat and it's not reserved seat. But it may be that in 50 years later, you can see that those are the switched to reserved seat. Because as you can see here, Korean society is aging really faster. So population may decline by 2050s. And as you can see here, the population by age, the portion of age over 65 is increasing dramatically. And by the year of 2050, maybe 40% of our population will be composed by people who is age over 65. And the speed of the population change is really fast. So about 2 years ago, out of the 20th century, the portion of senior citizen was number 10 in South Korea, and as time goes by, it will go up really fast. And for the other countries like Japan, it remains same, Germany, it remains same, but only Korea is speeding up. So society is really getting older and older. So it really burdens our society. So in 2005, to support the old people who is age older than 65, you require young people, maybe 8 people, but by 2020, you need 4 young people to support 1 old man, and by age 2050, you need one person to support old man. And the problem was that, as today's campus showed on the first day, the cost of health insurance is increasing to cover the health cost of old people. So by 2002, about 20% of our health insurance was spent just for old people. But at the age 2008, the percentage is increasing. About 30% of our health insurance cost is spent for old people. So here's the start of my talk, so genome hormesis. So as you can see, diet restriction, eating less, has been well demonstrated to extend lifespan in many animals. And as you can see here, calorie restriction by up to 55 or 65% increased the lifespan of mice. To have a meaningful lifespan extension, you need 30 or 40% calorie reduction. But the problem is that who really wants to do that? So enjoying those kind of food is really one of your life enjoy and pleasure. So scientists is now trying to find the chemicals that may mimic the effect of diet restriction. So you can go to buffet and take as much as food you like, and just take one pill, and the pill may mimic the effect of diet restriction, and your body can respond to that. So she's John Carman She survived 122 years. And before her death, she once told that her secret to her long lifespan was large consumption of olive oil and red wine. And scientists recently found that a compound called resveratrol is rich in red wine. So in fruit flies and in mice, people tested resveratrol. And so it was done in Mark Taylor lab. I was in his lab. So when feeding resveratrol, it increased the lifespan, and also the shorter lifespan of high-calorie diet-fed mice was increased by feeding resveratrol. And the interesting thing was that the action of resveratrol seems to be working in the same pathway of diet restriction. So resveratrol could be calorie-serving mimetics. So here's the general hormesis hypothesis proposed by David Sinclair. So plants, they are experiencing many kinds of stress, like UV stress, disease, attack by animals, dehydration. So in response to those stresses, they produce plant stress compound, and those kind of compound could be mild stress to your body, and by taking those compounds, you may extend lifespan. So I like to test other compound other than resveratrol, and the compound I've been tested was curcumin. So curcumin is rich in spice turmeric, and spice turmeric is quite rich in Indian curry. So diet turmeric has been well known for its function in Alzheimer's disease. So diet rich in curcumin and is found in Indian curry spice may help explain why the rate of Alzheimer's disease is much lower among the elder people in India compared to Western part. So people in India, they have really low incidence of Alzheimer's disease, and it may be due to their high consumption of curry. And when you also see the incidence of cancer, the people who does not take that much curry, they have higher incidence of skin cancer. But like Bombay and Japan, who people take lots of curry, they have low incidence of cancer. So curcumin may have effect on cancer and new generation and apoptosis, but it may also affect aging by touching those genes. But so far, nobody have ever tested the effect of curcumin on the lifespan in any animals. So we tested the effect of curcumin on fruit fly aging. And as you can see here, feeding curcumin extends the lifespan of fruit flies. And in addition to just lifespan, it also increases the health span. So it's done by measurement of negative geotaxis, and they are much more healthier when curcumins were fed. And also feeding curcumin increases the stress resistance. So as you can see here, oxidative stress, in response to oxidative stress, they are resistance. And we did microarray after curcumin feeding. So the panel 1 shows the gene changes by aging, so 1 day versus 40 days. So the genes like Methuselah, TOR, or insulin receptor, and the genes about Jun kinase, they are all increased by aging, but short-term feeding and the third one is long-term feeding, 40-day feeding of curcumin, it all reduced the gene expression language of those genes. So here, so the genes in the 1 day, and after 40 days, INR, TOR, Jun kinase 1, and Methuselah, it all goes up. But in the fourth graph, for curcumin feeding, it all decreased the age-related expression of those genes. And the next, the compound is not compound. The next extract is Korean mistletoe extract. So mistletoe is obligate semi-parasitic plant, and it is one of the most widely studied complementary and alternative medicine therapy in people with cancer. So in most European countries, it is much like Korean ginseng. And we fed mosquito extract in fruit flies, and as you can see here, in both males and females, the lifespan was increased by more than 30%. And it also increased the lifespan of C. elegans. And mosquito extract, we may work as calorie-stream mimetics. So it is 12% is our ad-libbed condition. So in ad-libbed condition, mosquito extract, it extends the lifespan, and 2% is our DR condition, and 2 is yeast diet. So 2% yeast diet, it extends the lifespan compared to 12%, but adding mosquito extract to the 2% diet did not further extend the lifespan of fruit flies, which means that mosquito extract may work as calorie-stream mimetics. And we also changed the several longevity associated genes, like feeding mosquito extract increased the expression of SORT2 and decreased the expression of AKT, which is one of the active components in insulin IGF-1 signaling. And interestingly, in addition to its anti-aging effect, it also touches the mobility or exercise effect. So by feeding mosquito extract, it increased the endurance exercise activity of mice. So it increased the time in treadmill and time in the distance of the treadmill. And also swim time was greatly increased by mosquito feeding. And second one is, second one I'd like to talk about is radiation hormesis. So here is the current situation in Korea. So about 20 nuclear power plants are in operation right now, and it will be increased by 28 plants in 2016. And about 40% of our electricity comes from nuclear power plant. So the generation capacity is number 6 in the case of Korea. And about 40% of our electricity comes from Korea. And now that we have small land, but we have lots of nuclear power plant, the number of power plant in one this area is number 1. So why we do that? Because we are a small country, we have little natural resource, and so we have to depend on the import of many natural resources. And because nuclear power is quite economical, we are dependent on so much. So uranium 1 gram, it equals to 3 tons of charcoal and 9 grams of oil. So to run 1 million kilowatt power plant for a year, you require just 30 tons of nuclear power. So 10-ton truck, 3 10-ton trucks. But for the charcoal, you need 100,000 tons of ship, and you need 22 ships. So it is really economical. The problem is that when there's something about nuclear power plant, there's a big riot in South Korea. So about several years ago, Korean government, they want to set up a nuclear disposal facility in one island, and there was a big riot, thinking that, yeah, they will destroy everything in the island. So there was a big riot, and eventually the plan was cancelled. So why we do that? Because, yeah, surely people fear about nuclear plant. So like Fukushima, yeah, people fear that, oh, what if the plant explodes? So that's the main reason. And another reason is that there's some fear about nuclear. So we call it nuclear phobia. And it's mainly done by news media. So it is news media in Korea, and I can translate for you. So here's the goat, and just in a village that is close to nuclear power plant, there was a newborn goat with malformation. And people think that, oh, what if, and they assume that it is due to nuclear leakage from the nuclear power plant. And also, there was a guard in nuclear power plant, and his wife gave birth a newborn child, and the child had no brain. And also, they claim that, oh, it may be due to nuclear explosion. So people always think that near the nuclear power plant, there could be some leakage, and it could be dangerous to your health. So Actually, the amount of radiation we are receiving is quite less in nuclear power plant. So it is the amount of radiation we are receiving from natural resources and artificial resources. So for the round trip from Tokyo to New York, you are receiving 200 microsievert, and surely you are exposing to many sources of radiation like space, food, earth, and radon. So actually we are receiving 2400 microsievert of radiation per year. And when you do a chest X-ray, you receive some amount. Surely CT, you receive more. And actually, the amount of radiation you are getting from nuclear power plant is minimal, like 50 microsievert per year. So why we have some fear? Because we do not know the response of low dose radiation to our health. So there are three models regarding to the effect of low dose radiation. So linear non-threshold model LNT is suppose that there is no threshold. So even low dose of radiation is detrimental to your health, and it may increase the radiation-related cancer risk. And linear threshold model claims that there is a threshold. So up to certain amount is okay, and if it is above the up to certain amount, it increases the incidence of cancer. And hormesis model is opposite. So the low dose of radiation, instead of is detrimental, it could be beneficial to your health. So we'd like to test the model of radiation hormesis by getting scientific evidence. So by getting the scientific evidence of radiation hormesis, we can reduce the protection cost. Also, we can improve the public understanding, and we can apply it for diverse medical use. So what is the evidence? First, as is essentially studied by Alex Moskalev, we also have seen that low dose of radiation at the egg stage, radiation at the egg stage increased the lifespan. So surely high dose of radiation, like 4 gray, it shortened the lifespan, but low dose of radiation, like 0. gray, it increased the lifespan with acute irradiation and chronic irradiation. So we measured the gene expression changes using microarray, and with about 1800 genes are changed, and 65% of them was upregulated. And we found that some genes that is mainly involved in the metabolism, like protein biosynthesis and carbohydrate metabolism, phosphate metabolism, those are upregulated. And when we compare the gene changes by ionizing radiation and dietary restriction, it is quite opposite. So ionizing radiation, protein synthesis is upregulated. In DR, it is downregulated. And DNA metabolism is up and it's down here. Stress response goes up. So surely it is really opposite, and there's just a few pathways that is overlapping each other. So we think that the lifespan extension effect by ionizing radiation is totally different from those of dietary restriction. And second, we'd like to see the effect of ionizing radiation, low dose of ionizing radiation on innate immunity. So it is known that through the many mammalian studies, it's known that low dose of radiation can activate immune response. But in the case of mammalian, the immunity is composed of innate immunity and adaptive immunity. And adaptive immunity and innate immunity, they are really connected to each other. And we do not know whether low dose of radiation can activate innate immunity or adaptive immunity or both, because they are connected to each other and they can stimulate each other. So we'd like to invest the radiation-induced innate immunity, because fruit flies has only innate immunity system. So here's the innate immune system, as really well conserved between invertebrate and vertebrate. And innate immunity can be divided into TOR pathway and IMT pathway. TOR pathway, it is activated by fungi and gram-positive bacteria. IMT pathway is activated by gram-negative bacteria. So TOR pathway and IMT pathway both activate antimicrobial peptide. So we first screened the drosmycin, which is Toll pathway activated antimicrobial peptide, and it's tagged to GFP. And we found that only at low dose of radiation, this GFP is highly expressed after low dose irradiation. And we found that after gram-positive bacterial exposure, flies start to die, and the death rate is much lower when they are exposed to low dose radiation. And when they are exposed to high dose radiation, they are quite susceptible. And it was also true for the case of gram-negative bacteria. So for gram-negative bacteria, low dose radiation exposure increased the survival of fruit flies, infected fruit flies. We'd like to see the effect pathway changes by low dose radiation, and found that several key component proteins are activated by low dose radiation. And the proteins that is involved in only toll pathway are activated, like dorsal and pele, it was activated. Surprisingly, the pathway that is activated by the pathway of the IMT pathway, it is not changed at all. So low dose radiation, as I've seen you, it increased the lifespan and it increased innate immunity. And due to the time limit, I did not show the data about tumorigenesis, but it also decreased the rate of tumorigenesis. So there we are getting the evidence for the radiation hormesis right now. So let me conclude my talk. So we are really have been searching for the fountain we use. So have we found it? Yes, we have found it. Yeah, we found it. So two of us is living daily, and you have to eat less, drink wine, and eat curry. So my work is done by many members in my lab, and University of California, Chapari, she collaborated each other with me about the curcumin project, and Dr. Lee from CRIP, he's also helping me with microarray, and Handong University, Dr. Kim is helping me with mistletoe extract, and Radiation Health Research Institute, he's helping me with radiation project and funding source from the Korean Research Foundation. And I'd like to show the presentation to the next IAGG meeting. Oh. Yeah, so it's 20th IAGG World Congress. So it will be held in next year, June, and COEX in Seoul. And about 60,000 people is expected to attend. So there are 6 keynote lectures, and 2 of them are biological science, stem cell aging, and signaling in anti-aging process. So there are 4 categories of presidential symposia: clinical medicine, social science, behavioral science, and research, policy, and practice. And I'm organizing the symposia for the biological science. So we have 10 biological science presidential symposia, and all the speakers is already selected for the presidential symposia. What you can do is that you can submit your own symposia. So submission is already open. The deadline for submitting symposia is end of May, and we are going to review it, and we are going to notify the acceptance by the end of September. The good thing is that you can enjoy Korean culture by staying in our home country. Yeah. So we are experiencing homestay program. So providers, the members of our society, and citizens, some people from developing countries and students, or people who want to experience Korean culture, they can participate. And the expense is free of charge or minimum expense, like $30 per day. And you can see they are wearing Korean traditional clothing and speaking each other. So key date, call for the symposia by end of May, and call for the oral and poster abstract is end of October, and homestay is next year's May. So we should come here, so meet me with Korean history and culture. There are 4 UNESCO World Heritage in Seoul, and there are many royal palaces, many museums and art galleries, and traditional housing like that. And meeting with nature, so there's beautiful stream, Mountain Namsan, beautiful forest. And you can experience Korea, like taekwondo, Korean wave pop, or tea making, tasting traditional food, temple stay. Actually, if you want, you can stay in temple. And you can experience the making Korean food. And enjoying entertainment nightlife. There are many casinos and observatory, traditional market, club street. And other tourist attractions like Korean battlefield tour, like DMZ, demilitarized zone. You can see North Korea from there. And many festivals out there. And shopping is great. There are many places to go out for shopping. And enjoy the cuisines, so you can enjoy delicious food there. So I'd like to welcome you to the next year's IEGG, and if time permits, yeah, I prepared a short movie. So I hope I can see you next year in Seoul. Thank you.
Well, well done for the announcement of the next Congress. In your biology section, I will suggest that the topic 5, you still have written the old obsolete words, stress response and homeostasis. Please, if possible, change it to our modern understanding of biology, which is homeodynamics. Scientists are one of the most conservative group of people. We don't change that fast, but we are the ones who are supposed to change with the new knowledge. So that will be nice. But because we are respecting the time limits also, I don't know whether we can go into some discussion, any questions. You are using 2 hormetins, one physical hormetin radiation is a good example, and a nutritional hormetin from mistletoe, which is another good example in the showing that yes, it works. Mikhail, you have a question? Okay, go ahead.
Did you use feeding assay for Drosophila when study effects of curcumin?
Yeah, that's a good question. So whenever we test any compound or plant extract, there should be some criteria. So we try to see whether the longer lifespan by feeding the compound or plant extract is not due to side effect like lowering the metabolism or by reducing the feeding rate. So if the compound or plant extract reduces the feeding rate, it may mimic the effect of diet restriction. So our anti-aging compound is all compound by feeding rate, and it does not change the feeding rate at all.
What quantity of curcumin have to eat human to increase lifespan?
Oh, it's really tough question because it's really hard to measure the feeding rate of fruit flies. And right now, NIA, there's an intervention testing program called ITP. And ITP is testing the effect of curcumin in mice. And the concentration that is testing right now is 4,000 ppm. And it was started in 2007, and I expect it will be published soon, maybe this year.
Well, that is always the biggest question, what we do in the experimental systems and what can be taken from food.
Usually we use very low concentration, but may be pharmacologically used in human.
Just as a technical question, how did you measure the feeding rate?
There could be several methods. One is called CAFE assay. So using small capillary tube, you can see how much they are feeding. What we are doing is that we use blue dye. So using bromopenol blue, actually we can see the blue dye ingestion, and the amount of blue dye ingested can be actually quantified by using spectrophotometry assay.
Yeah, I think I'll ask you later about the details.
So in the Korean food culture, what is mistletoe used for normally as a?
We usually use it for treating cancers, and also they have good effect for the diabetics.
All right. Then we come to the last speaker of the session, unless Marina is here. No. So Mikhail Shpesnikov, you have to wind up the session now.